Interesting. Let's see.
Good morning, everybody, and happy Friday. Welcome to the Friday Informatics Institute PowerTalk seminar series. I'm very happy to introduce today's speaker, Dr. Elliott Lefkowitz. And I hope I'm pronouncing that right. He's very much well known to us, professor of microbiology at UAB, also with additional appointments in computer science and in genetics, and multiple center appointments in AIDS, musculoskeletal, arthritis, cancer, translational research, and also in our own Informatics Institute. And he's also director of the CCTS Informatics Program and the Molecular and Genetic Bioinformatics Facility in the Center for AIDS Research, also director of the Bioinformatics Core and the Microbiome Facility and the Viral Bioinformatics Resource Center. His interests are in, research interests are in microbial genomics and evolution, and also bioinformatics and the microbiome. And this work was especially important during the COVID pandemic, as all the virology and the genomics of that. And he's also co-director of the UAB undergraduate bioinformatics program, which has been wildly successful. The name of his talk is Virus Surveillance, Tools to Classify the Unknown.
So thank you, Amy. And as the title of my talk suggests, I'm going to concentrate on some of the more basic research interests that my lab has, and talk about work that we're doing under the auspices of an NIH grant to support information about viruses. And I'll talk a lot more about that and what that means in a bit. First, and as always, I'd like to thank the people who are involved. Heavily involved in the work I'm going to be talking about is Don Dempsey, a programmer analyst in my group. Don's been with me for almost 20 years now, since 2003. Curtis Hendrickson also is involved in much of the work that I'm going to be talking about. Curtis hasn't been with me quite as long as Don. He started in 2005, so long enough. And then more recently we have Alexei Shipanov, who's been helping us with the work. And then I'm going to show you work related to some of the information that we're providing on different viral families. And Donald Smith has been working with us on that. And I'll mention a few others along the way. Okay, so this all starts with feeling sick. And a lot of us have experience with this in the past couple years, but pretty much we've all experienced being sick for our whole lives at one point or another. And so what happens if you're sick? If you're sick enough, you'll go into the physician's office and you're going to ask the question, and especially they're going to ask the question in order to treat you, what is it that's causing whatever ails you? And so frequently that may be due to some microbial agent. And so in that case, based upon the symptoms that you have, you want to start to look and you want to find out what it is, what bug it is that's causing your illness. And finding that can tell you a lot about the prognosis, but certainly what you're interested in and what your physician is interested in is how you treat it. But that's the first step, finding the bug. So you can go about that in any number of ways. The physician has a set number of laboratory tests that they run. But for something like SARS, originally back in 2003, certainly with SARS-CoV-2, starting in late 2019, the agent was unknown totally. And so it wasn't clear what was causing these illnesses that started to pop up rather rapidly, initially at least. And so what you do, you start doing basic detective work. And if you suspect a viral agent or even a bacterial agent, you might try to isolate that. And so you might get electron micrograph, for example, of a picture of the agent that might be present, might be responsible, might not. You need other data, but you might end up with a bunch of EMs like this. And so, okay, you stand back, you look at these and you say, which one of these might be responsible for the particular symptoms that I have? Now, for us as bystanders at this point, at least, we might want to ask, which one of these would I least want to get? Which one of these wouldn't I mind getting? And what you see here, you see some pox viruses over to the right. These are coronaviruses to the left. This one might not be the bad one to get in the upper left here. That's mouse hepatitis virus, which pretty much would not infect a human being. Whereas down here, a number of us have gotten this over the last 2.5 years. This is SARS-CoV-2 that's responsible for COVID-19. Here we have the pox viruses. This one here is monkeypox, which is responsible for the current outbreak. I might not mind getting that too much. I'm vaccinated against smallpox, and so I should have a rather mild illness. But many of you, certainly any of you born after around 1980 or so, have not been vaccinated against smallpox, and therefore you could get a fairly significant illness, certainly almost not a fatal illness given the current monkeypox strain that's that's infecting people around the world these days. What you don't want, especially if you haven't been vaccinated, is down here, this virus here. This is variola virus. This causes severe disease with significant mortality. And so certainly if you haven't been vaccinated, you won't want this. I don't want this. My vaccination status is probably waning sufficiently that I would get sick to some extent here. These up here are animal pox viruses. This is Orf virus that infects goat and sheep. This is Yatapox, which does infect primates. So probably I would choose this one here, which probably at best would give me a little wart-like lesion, and that would be it. Okay, but if you see these in the electron micrograph, you see that you'd be hard-pressed to tell the difference between all of these pox viruses. You'd be hard-pressed to tell the difference between these coronaviruses. So just looking at the agent in itself doesn't tell you much in terms of how serious illness you might have. And of course, what we do today, the very first thing that was done when people started getting sick with SARS-CoV-2 is you took the samples that you had and you sequenced them. And you do metagenomic sequencing, you sequence everything that's present. So you get a lot of human DNA, of course, that you filter out, and you see what's left. And you see if you have a sequence that's reminiscent of a pox virus or a coronavirus, or something totally unique like this thing down here in the corner. This happens to be a archibacterial virus. So you pretty much don't have to worry about this if you get this, because it only infects archibacteria. But you don't know what you're going to get until you actually do the sequencing and see what's present. Of course, finding an agent doesn't mean that that's what's causing the disease. You need other information in order to make the direct association with disease, but at least this gives you something to investigate. But okay, we have the sequence. What do you do with that sequence? And I'm sure pretty much everybody listening on my talk would give the answer that the first thing you do is you take the sequence and you blast it against GenBank and you see what's there. And so if we had a sequence of monkeypox and we blast it against GenBank, you may be able to see here that the hits, always lose my cursor in these things. Come back, cursor. Okay, here we go. You see the hits are all to monkeypox here. So pretty much that gives you an idea of what's present in that population at least. If it was something that was related to monkeypox, but not closely related, we might see other hits here. And what we do is then let's say we really weren't that familiar with monkeypox and we didn't know anything about the disease it would cause. So what we come down here, we can look at the taxonomic relationships based upon the blast results that were returned. And again, it shows us monkeypox. And if you click on the organism itself, what you get is the GenBank record. And what's important here in terms of what I'm going to be talking about today is the organism and the taxonomic description of the organism, which is provided here in every GenBank record. And so we see here that this says monkeypox. We'll click on that in a minute. But this taxonomic relationship is something that NCBI doesn't determine. It wasn't determined by the person who discovered monkeypox. The taxonomic relationship, as we'll see, was determined by the International Committee on Taxonomy of Viruses. And that's going to be my main topic of discussion today. We look at the record, as I showed you here, we look at the record for monkeypox, the taxonomic record at NCBI, and you see that full taxonomic lineage. We see that monkeypox virus is a particular species in the Poxviridae family of organisms. You can also, of course, get access to all sequence records. We come down here at the bottom and we see all of these links down here at the bottom. And all of these links tell us give us access to different types of information, but a lot of these are taxonomic information. So we see here we can get taxonomic information from this Encyclopedia of Life. We can get it from what is it here? The global, let's see if I can, I can't see it here. The GBIF is what this is. This is another database of taxonomic information. We can also get it from Wikipedia. But all of the taxonomic information, whether it be from NCBI, whether it be from Encyclopedia of Life or GBIF, all of that comes from one source, and that is the International Committee on Taxonomy of Viruses. So if you click on that, you'll come to the sequence record that's provided by the ICTV. So what I want to do now is describe what the ICTV is, what it does, and how the work that I'm going to talk about in detail relates to the activities of the ICTV. Okay, so the ICTV is the International Committee on Taxonomy of Viruses. The URL that you can go to is ictv.global. We try to make that pretty simple. And so the ICTV determines the taxonomy of viruses. And taxonomy is a fairly simple concept. What it means is classification plus nomenclature. So we take all of the viruses that are in existence that we know of, and we classify them according to various principles, and then we classify them into taxonomic ranks. And I'll talk more about what the ranks the ICTV uses are. But then we name the ranks according to its constituents. So that is the species that belongs to monkeypox. We've named the viruses that belong to that species monkeypox virus. Monkeypox virus is categorized into the genus orthopox virus, and orthopox virus also contains species such as variola virus that causes smallpox, ectromelia virus, which causes smallpox, vaccinia virus, which is used to vaccinate people against smallpox. So all of those species are categorized into the genus orthopox virus. And so that's classification and nomenclature. So why does the ICTV do this? Well, the ICTV has a specific charge by the International Union of Microbiological Societies, the Virology Division, to define, refine, and maintain a universal virus taxonomy. And it started doing its work back in 1966. And so it's been operational for 55 plus years. Its first report on virus taxonomy, classifying and naming all the viruses known at that time, came out in 1971. And since then, 9 hard copy volumes have been published, the last in 2011. In 2016, we went online. And so now the ICTV reports that provide all of the information on the current viral taxonomy is online at that ictv.global site. Okay, but why do we spend so much effort determining the viral taxonomy? So taxonomy in general, and certainly virus taxonomy, allows us to understand biological organisms, according to their relationships with one another. So for an example, we can look at a gene in mice. And in mice, that gene has someone's done functional studies and you know what that gene does. And in humans, someone may be working on some disease and they found an association between a human gene and that disease. So they sequence the gene, they then look at it in GenBank and they come up with that mouse hit, and that mouse hit has some sequence similarity between humans. And so we know that there is a taxonomic relationship at the level of mammals between humans and mice. And so we might be able to then take that functional information on the mouse gene and use that to infer a similar function in humans. And now we can begin to test and see if that function holds, see how that function might relate to the disease in humans. So by looking at those, by understanding the relationships between organisms, and then looking at the similarities and distinguishing properties between the organisms and between the genes of the organisms, and whatever it is we happen to be studying, we can now begin to infer properties in one organism that we haven't yet studied in detail, but infer properties from an organism that we have much more information about. And by doing this, we can begin to build a much more holistic understanding of the biology of all organisms, being able to better understand more closely related organisms to some reference type organism, but using that to infer, again, the properties of other organisms that we're just beginning to study. Viruses are important, of course, because they infect, in essence, every organism on the planet. They have symbiotic relationships in many instances in which they help maintain the ecosystem. But as we know, they can also cause disease. And so viral taxonomy itself is no less important than the taxonomy of any other biological entity, because by classifying viruses and naming them, we can now make those inferences between different viruses when we find something unknown, but through sequencing, we're able to match it up with something that is known, and then we can look at common properties between those two organisms. So the ICTV, as I said, has been operational now for 55 plus years, but we still closely look at the properties that are used to help define the viral taxonomy. Back in April of this year in Oxford, there was a meeting convened in which the topic was virus classification in a metagenomic age. And so a lot of the principles that came out of the meeting have been principles under which the ICTV has been working for decades, but they help refine the principles that we're using while incorporating viruses discovered as a result of metagenomic sequencing of different environments, whether it be seawater or whether it be environments anywhere that you can think of. And so a key principle of ICTV taxonomy is that the viral taxonomy needs to reflect the evolutionary history of the viruses themselves. And so a lot of that, I'll talk about some of the characteristics that are used to define the taxonomy, but a lot of that does come from sequence information and reconstructing the phylogeny of the viruses. But when you do taxonomy, you also use the property of the organisms themselves. So virus properties are used to guide the classification of the viruses into different ranks. But still, the guiding principle holds that the evolutionary history of the viruses is the key indicator of the correct taxonomy that you're trying to build. Now, taxonomy based on evolution is only one possible taxonomy. One could also imagine a taxonomy based upon host. And so viruses that infect the same host may have similar properties that are different from viruses that infect another host. Other properties of the viruses may also guide a taxonomy, the disease that they cause. So viruses that cause a respiratory disease may have unique properties that distinguish viruses that cause a disease in the neurological system. And so all of that can be important. It can be more important to different organizations. The CDC and the World Health Organization, for example, may be more interested in disease that viruses cause. The US government and other governments around the world may be interested in classifying viruses according to their potential for being used as a biological warfare agent. So all of those are important, but the ICTV still has to have a solid basis for its classification. And so that still comes down to the evolutionary history of the viruses. And so when we look at the classification of viruses, this is just a pictogram of different viral families that cause disease in humans. And so this is sort of a comprehensive indication of the different viruses that cause disease in humans, but we can use it to look at the different properties that are used for taxonomic classification. And so phenotypic properties, as I said, are still important. It could be the composition of the genome, whether they're DNA or RNA, whether they're double-stranded DNA viruses or single-stranded DNA viruses plus sense, meaning they can be directly, the genome can be directly translated into protein. Whether or not it has an icosahedral capsid, like these viruses up here, or whether or not that capsid is not icosahedral, has more of a general amorphous type appearance, like those for Poxviridae. The host range can be important, and disease, whether or not they cause disease in certain organisms, are certainly important and can be taken into account during classification. And these were the only properties back in 1966 that were available in order to classify viruses. So the initial ICTV taxonomy was built on phenotypic properties. But of course, these days, we now use genotypic properties, the gene content of the genome, the order of the genes in the genome, how they're expressed over time, given the virus's life cycle, but certainly sequence similarity and using sequence similarity to do phylogenetic reconstruction is what many of the study groups in the ICTV that work on the different classes of viruses, that's what they use these days. But if you look over the years, if you compare the phylogeny that was created with phenotypic properties to the phylogeny that, well, the taxonomy that was created with phenotypic properties to the phylogeny of the evolutionary history of those viruses, you see it worked out rather well. There certainly have been changes over the years, moving some taxa from one higher rank to another higher rank because of the different properties. But in general, what we had in 1966 holds up to what we currently have in 2022.
Can I ask you a question?
Yeah.
So there's well, maybe you're going to talk about it on the next slide. Go ahead.
Okay, these are the different ranks that I'm going to talk about that the ICTV uses for its classification. And you can see here there's seven major ranks with sub-ranks as well. We start at the level of species. So individual viruses are classified according to common properties into species. Those species that have common properties are classified into the same genus, and there are multiple genera. And then you go on up to families, orders, class, phylum, kingdom, and realm.
So my question is, does this make sense for viruses? Are they clearly, is there a clear demarcation between when a species becomes a genus, or there's things in between? And then there's the variants, right, which are subspecies in effect.
Right.
So does it make sense? Is there a clear delineation where you can, like if it had a different number of chromosomes, you go, okay, there's some big difference there, but it seems like a continuum to me.
So I'm going to talk a lot about demarcation criteria and how you
also, just a quick comment. When you ask questions, please speak up so we can hear you. And can you just summarize that question really quickly so that in case we couldn't hear? That's okay.
So Jim was asking, what are the criteria that are used to classify viruses at these different ranks, and whether or not the criteria are continuous, or are there discrete criteria that can be used to make a objective classification of a virus into a particular rank. And I'll come to that, and I'll talk a good bit about that in just a little bit. You get a sense for what those criteria are when you look over here to the right. And what you see here are criteria that are used at different levels of the taxonomic rank hierarchy. So at the lowest level, when we're talking about species, genera, and families, we're looking at nucleic acid sequence identity, amino acid sequence identity. When you're looking at the family to core ranks, you're bringing in core genes that are present, that are common to the viruses at those core ranks. At the higher levels, going anywhere from the phylum on up to the realm, we're looking at common functional motifs and common structural folds, because at these higher levels of taxonomy, the level of nucleic acid identity and amino acid identity really has fallen sufficiently that you can't classify at these higher levels. Because if you think about it, viruses infect all forms of life on Earth. Many viruses have probably been around since life itself evolved. So we're probably talking about multiple hundreds of millions of years, if not billions of years, when we distinguish viruses at the realm level. And I'll talk a little bit more about that in a number of slides. Just to show you, we distinguish different realms, different ranks, by the suffixes that you add to the name. So nomenclature is particularly important. All genera end in virus, whereas all families end in viridae. Species, the ICTV has just adopted a binomial name nomenclature in which the first word of the species is the name of the genus, and then there's a second word that's unique to that particular species. Okay, so that is what the ICTV does. Now, why am I here talking to you about the ICTV? So I started as data subcommittee chair back 15 years ago with the ICTV. My position is now data secretary. And so the role of the data secretary is, and this is in the statutes of the ICTV, is in essence to provide the public facing point for the ICTV. So all of the information that we generate, which especially and primarily is the taxonomic classification of the viruses, All of that needs to be provided to the public, whether it be the scientific investigators, whether it be government entities, whether it be just interested public. So we want to make that available in one form or another. We also want to provide all of the reasoning behind the decisions that the ICTV makes. We also would like to provide tools that allow people to drill down and get more and more information about the viruses and their classification. So back when we started, when I started in 2007, there was no public ICTV website. There was an Excel spreadsheet that was distributed with the taxonomy, but there was nothing more than that. So with the help of Curtis and Don and a few others, we developed the initial ICTV website. This had some funding from a few grants that I had back then in NIH. But over the years, when I didn't have grants specifically directed at virus properties and virus taxonomy, we did this pretty much as a sideline on our own time, developing tools over the years. But there was always a lot more that we wanted to do. So we could always provide the basic information. That took a bit of work, but we were happy to do that. But we knew we wanted to provide more information, more tools and such. And so a couple of years ago, there was this NIH funding opportunity called biomedical knowledge bases that function to take information about biological entities and organize it, annotate it, and make it available to investigators around the world. And so I submitted a proposal, virus taxonomy, a community knowledge base supporting virus research under this biological knowledge base RFA. And about a year ago, that was funded. And so with that, we had a number of different aims that we've been looking at. One, we wanted to ensure that the infrastructure supporting the work of the ICTV website and its communication was solid and was able to hold up years into the future. We wanted to add as much information. We wanted to enrich the information available on virus taxonomy and the viruses that are being classified. We wanted to develop a number of tools that allow people to either support classification or at least support the ability to find the viruses that they're interested in, find what their classification is, and use information about those viruses in their own work. All of that needs to be accessible. We can create the greatest set of information and tools in the world, but it's worthless if it's not available to people. So we wanted to make it accessible in any number of different formats. And then we wanted to let people know about it. We wanted to provide the outreach and the training so people can utilize our resources. So that's what the grant is doing. So now, over the last year, we've completed our initial efforts on the grant. The primary one was to migrate it to new infrastructure. So before, it was run on a server here at UAB, even though people contributing to the effort are from around the world and people around the world access that information. So we moved it from that server to a server based on, in Amazon Web Services, AWS. We migrated that. We also changed it from a proprietary Microsoft-based system to the open source Drupal system that runs on Linux. So all of the data that we had, all of the databases, all of the tools that we had to visualize the taxonomy, all of that had to be migrated. And so that took upwards of 3/4 of a year or so in order to do that migration. And then we had to enhance some of the tools that we were providing. And in the middle of all this, there was a new release of the taxonomy. The ICTV releases a new taxonomy every year. But we were able to do all of that and come up with the website that you see here. And so I did want to just go to that website and show you a little bit about what that website can do and what type of information is available on it. I can find my cursor again. I can reshare. Cursor here. There you go. Okay. Here and share here. Okay. So back up here.
You have a link on that page.
A link.
The whole screen, you can just
Yeah, I just want to hide this thing here. So floating media controls. There we go. Okay, there we go. All right. So this is the ICTV website here. And this website is where you come in when you go to ictv.global. And so we have lots of different things here, news, a number of the resources that we have available are highlighted here. Importantly, if you come to this and you go to the taxonomy browser here, this is where you get access to the complete taxonomy. And so this is provided in a tabular format here. If you mouse over any of these categories, you can't see that. Oh, I know. Sorry about that. I think I shared the wrong thing. Okay, let's come back here and share again. That's the problem. I had the website up on the PowerPoint and it looked just like the website that you're seeing there. I'll do what you suggest. Yeah.
Slideshow.
Yeah, well, okay, I'm gonna have to stop that. Okay, there we go. All right, now we'll reshare.
I think there are two screens.
No, I do, but when I share, it'll work. Okay. It's just that PowerPoint took over. Oh, come
on. We are seeing the website, the web browser.
Yeah, I'm not seeing it.
Okay. So you are seeing, okay.
Okay.
Yeah, which is now going to make it more difficult because it's got out. Okay, so this is what I was talking about in terms of the taxonomy browser. And so the taxonomy browser provides access to the complete ICTV taxonomy. Given we have all of those 7 ranks and sub-ranks, it can be hard to find what you're looking for. And so let's say, for example, you wanted to see what the species related to monkeypox are. So you wanted to find monkeypox. So we can come to our search screen here.
Actually, for some reason, it's really enlarged. So I don't know if, so we're only seeing a part of it.
You're only seeing a part of it.
Like the upper left part of the screen.
Just a second.
And it's not live. We don't see the search.
I'm sharing the screen. Just a second here. So you still see only a part of it? All right, wait, I'm going to stop sharing then.
We just see what looks like a frozen screenshot of it.
Now, do you see me?
Yes.
Good. Yes, hi.
All right, just a second. Okay, now do you see the web browser? Hello?
It says you have. It says you started screen sharing, but it's blank.
Okay. Sometimes. Tell you what.
But it takes guts to do live demos.
Yeah, and just a second.
It sometimes helps when you share is to share your desktop and then you have.
Yeah. Please, I can do this. Just let me. The windows are getting.
Okay, now it looks like a web browser.
Yep.
Yeah,
like
and what I'm going to do is I'm going to mirror my display. And that, okay. Okay, how does that look?
That looks good.
Okay, good. So what I was going to say is, here's the taxonomy browser here that allows you to drill down, and you can open any of these and you can start to see what taxa might be present. Yeah, okay.
I'm just trying to monitor.
Okay, there we go. But it's hard to memorize what all these upper level taxa are. And so luckily we have a search form that you can come in. I can type in monkey and immediately I'll get a list of all the viruses that have, or all of the taxonomic ranks that have monkey as part of the name. And so we can come along there and we can see here that monkeypox virus is down here. So I can click on view here and it now will automatically take me to monkeypox. And I can see that monkeypox is a member of the genus orthopox virus, which contains all of these other species here. And so that's the taxonomic browser that can give you access to all of the information in the ICTV. This is the current release here. Let me reset this. So the current release 2021 covers 10,434 species, and we have 6 realms. Importantly, you can ask, what is a realm? And a realm, in essence, is defined by a unique origin of viruses. So each realm defines a class of viruses that has a unique evolutionary origin. So unlike all other forms of life, whether it be bacteria, eukaryotes or archaebacteria, which have one common origin, one common ancestor, the evidence is that viruses have multiple origins. And so that's reflected at the level of the realm. There are other viruses down here that you see that aren't yet grouped in a realm because we don't have sufficient information to classify them into that realm. You also have information on all past releases. So we can come down here and we can look at the release from 1971. And there you see only a couple families were defined: picornaviruses, which holds polioviruses, papovaviruses here, which are small DNA viruses, papillomaviruses, and polyomaviruses. So this is what the state of taxonomic classification was back in 1971. Importantly, what I wanted to show is that in addition to the taxonomy itself, we have information available about all of the different taxa that are classified. Now, not all of it is yet available. What we do is we compile this is the ICTV report that used to be in a hard copy, but is now available online from the website. And so using these controls, let's say we're interested in single-stranded DNA viruses, we can click on that and we can immediately see all the single-stranded DNA viruses. If we click on one of the families, now we get the report information, which contains a significant amount of information at the different taxonomic levels. So this is describing the family of viruses, Geminiviridae, which are plant viruses. If we want to look at any particular genus, here we see the particular information about that genus. It also has the demarcation criteria that I'll talk more about that define the viruses that go into that genus. And then it provides a list of all of the member species that are part of that genus. Okay, so let me go back now and see if I can get us sharing the screen properly. Okay, good. Now that's showing up. So can you see my last slide? It's the websites all go on down. So now you should see.
Actually, we're not seeing sharing. We're seeing you, but not the sharing.
Okay, just a second. I think it didn't help that I didn't share that. So let's see.
You shared it to some other place.
Here you go. Now you should see it. You see it now?
No, it's blank.
Oh, it's paused again. Wait a minute. Sorry, you gotta. Okay. Now let me do this.
Oh, there we go. Slide share. That's good.
Okay.
Oh, and actually, we're seeing your other screen. We're seeing your one with the next slide.
Yeah.
That's what happens when I messed up how I got it set up originally.
Also, when you interact with a strange computer.
No, this is my computer. It's just doing weird,
It's when you click on use slideshow, you should get the page.
the slide.
Okay. Let's do this. Stop mirroring. Yeah. And now we go. Okay. You see it now?
Perfect.
Okay, good. All right. So I talked about all the current efforts that we've been working on, and pretty much those are reflected in the website that I just showed you. So now I want to talk about some of the other ongoing projects that we have. And Jim asked the question, what distinguishes one taxon rank from another? How do we categorize viruses or groups or other ranks into all of the different ranks? And the way that's done is with demarcation criteria. So demarcation criteria are the distinguishing characteristics, whether they be phenotypic characteristics or genotypic characteristics, that allow us to compare different viruses and determine whether or not they can be classified together into the same species, same genus, or they require classification into different species or genera and such. And so in many forms of life, when you look at it, bacteria, for instance, there's a common demarcation criteria that's pretty much used for all bacteria. That is, if they have a level of nucleotide sequence identity that's 97% or higher, they're classified into the same species. For other forms of life, there are other characteristics that are used. But for viruses, we can't use a common set of characteristics for each group of viruses, because viruses, as I say, may have multiple origins, and different viruses, for example, double-stranded DNA viruses, especially double-stranded DNA viruses, evolve much slower than RNA viruses. And so the characteristics that you use for DNA viruses may be very different than you use, like percent sequence identity cutoffs. They may be very different. So the ICTV is composed of multiple study groups, one for each virus family, and they determine the demarcation criteria that are specific to the group of viruses that they happen to be experts in. In addition to looking at the phenotypic characteristics, when you look at the genotypic characteristics, there are a whole variety of different tools that different study groups use to determine the relationships between the viruses they're working on. And so all of these form part of those demarcation criteria. As an example, this is a tool called PASC, Conserved Core Genome. And what you do here is you just do a pairwise sequence comparison between every virus that's a member of a particular family, or every virus that you want to compare to look at their relationships. So you do a pairwise sequence comparison, you determine the percent identity between every pairwise comparison, and you plot it. And so these are the number of sequence pairs on the left axis that have a particular pairwise identity. That's, on the y-axis is the number of sequences in terms of percentage that have a particular percent identity along the x-axis here. And what you see is you see distinct peaks. You don't see a continuum in many cases. And so when you have distinct peaks like this, a peak out here can represent viruses that are in the same species that you're comparing against one another. Different peaks here in this range, this is for pox viruses, these represent different viruses that are categorized into the same genus when they're anywhere from 40% to 80% identical. Whereas viruses that are 20 to 30% identical belong in different subfamilies. And so this just shows that it's possible to have discrete values that you can use when you're creating the demarcation criteria for different viruses.
So does it occur that virus A has a 98% overlap with virus B, and virus B has a 98% overlap with virus C, but virus A and virus C have only a 96% overlap, say?
So Jim's asking, if you can have overlaps that in essence are showing different levels of sequence identity. And therefore, virus A and B may look similar when you're looking at, let's say, the left half of the genome, whereas virus A and C may look similar when you're looking at the right half of the genome, and virus B has no relationship to virus C with the right half of the genome. And yes, that can happen. Certainly, it could happen due to recombination. It could have happened due to recombination at some point millions of years ago. So you're looking at the true evolutionary relationships, but they've diverged quite a bit in certain regions of the genome. Bacteriophage do that all the time. And so when you're using a tool like PASC, you have to first define what regions of the genome happen to be important to demarcate virus, that particular group, that particular family. So every study group is going to define the parameters that have to go into a tool like PASC or any of these other tools that they're going to be using. Okay, so this is monkeypox. This is what was defined. These are the particular ranks that monkeypox are in. And then we look at the demarcation criteria at these different levels. We see that at the level of the realm, that is the level of the unique evolutionary origin of poxviruses and all of the other viruses they're related to, what's in common is the structural features of a protein that serves the function of a capsid protein. That is, it encapsulates the genome of the virus. And so if you have a structural feature that's a vertical jelly roll capsid protein, that allows you to place that virus in this particular realm. At the kingdom level, you get a little bit more specific in terms of that structure, a double jelly roll major capsid protein. At the phylum level, then there are other features here. You can look at the structural features that you see here, and you can see conservation between all of these different families of viruses and some of the pox viruses here that are used to classify them. If we look more in detail at those demarcation criteria, we have the phenotypic criteria for pox viruses. We're looking at orthopox viruses and at the genus level, pox viruses, Poxviridae at the family level. We're looking at host range. Pox viruses tend to be fairly host specific. We're looking at growth in tissue culture. We're looking at the disease characteristics that they cause and also serological characteristics. All of these can help define those relationships, but in the end, it's the genotypic ones. And so the amino acid sequence identity of a particular family of proteins coded for by the viruses can be utilized. Percent cutoffs for nucleotide sequence identity, defining differences between species in the orthopox virus genomes are used. Gene content, genome organization, the order of the genes in the viruses, and then finally phylogenetic analysis. So all of those come into play in order to allow you to create an evolutionary tree such as this that shows the different species here at the tips of the branches, and these are divided up into the different genera. And so that's how the taxonomic criteria come together to allow you to demarcate pox viruses. And then here we have monkeypox viruses in the genus. So the question is, what are the demarcation criteria? I just showed you the ones for pox viruses, but where do you find those? Well, there is not one place to find them. There are actually multiple places. They appear in the proposals people submit to the ICTV for new taxonomy. They appear in those report chapters that I showed you with the online ICTV report. They may appear in published transcripts, or they may be in someone's brain. So one of our major goals working on this grant in the next year or two is to develop a comprehensive database of all of those demarcation criteria. Another major goal we have is to have a virus lookup tool according to names. So someone comes in, they ask a question all the time. What's the species for polio? Is it poliovirus? Well, actually, if you look at the current 2022 taxonomy, poliovirus doesn't appear there. And so we want to make a tool that allows you to input the name and get back what the formal taxonomic species name and higher rank names are for the name that you're looking at. So what we do, we start with a database or databases of different names. And so these are different taxonomic databases, including NCBI, and they all have associated with them common names. But there's a lot of overlap, but there's also a lot of unique names in all these databases. So Don Dempsey in my group has developed a name annotation pipeline that has as input or has as a data set all of those databases or some of those databases that we can use to search against when someone inputs a name. So for example, if someone inputs poliovirus as a name, they'll find that poliovirus has other names associated with it, poliovirus type 1, human poliovirus 1 or 2 or 3, poliovirus, or human enterovirus C. And then by linking those database names to the ICTV taxonomy, we now know that enterovirus C is the species name for poliovirus. And so we're still developing that tool, and hopefully that will be available in the next number of months. One can ask a corresponding question. Let's start with the sequence of a newly isolated virus. And we want to find out whether that newly isolated virus is a member of an existing species or might be a member of a new species that needs to be created. So a graduate student of mine, Scott Halco, is working on this. He's looking at two approaches. One is a BLAST approach, in which we create a database of exemplar viruses, one for each species in the complete ICTV taxonomy, and we just search the newly isolated sequence against that database sequence and sees what hits best. But another approach is to use a more sophisticated alignment-based search, in which you take the newly isolated sequence and you align it to an existing alignment of family-specific viruses. And so if you have an alignment of family-specific viruses, you align your query sequence to that. And in addition, you have a scoring scheme that scores highly regions that are somewhat variable, in which you have maximal taxonomic information. Regions that are extremely variable don't allow you to distinguish one species from another. Neither are regions that are completely identical. So those regions that are too variable or regions that are identical have low information content. The other regions that are just right have variable information content, but not too variable. So he can use these now to score the alignment of the query sequence against these MSAs. And he's now experimenting with different methods of doing that, seeing which one is best, which scoring algorithm is best. And then finally, we're developing other applications to visualize the taxonomy, not just that row-based one I showed you here. So one method is to use these data-driven displays. And each one of the nodes here is clickable, so you can expand or contract that node. And Siddharth Kumar in the Department of Computer Science is working with me to develop this visual display, along with his students are helping out on this. So that's what we're currently working on this year. Over the subsequent years of the grant, we have a number of other efforts that come into play. We want to enhance the infrastructure, enhance the performance, because right now, as I said, we have 10,000 species. We're now categorizing viruses for metagenomic sequence information, and we fully expect to have hundreds of thousands, if not millions of species in the future. We want to add as much information about the viruses that have been classified to the databases and make that available to people. And we want to enhance further the tools that are available. Right now, we have a manual process where people create on an Excel spreadsheet and a Word form information that they need to propose new taxonomic classifications. What we want to do is automate that whole process, whereby the quality control of the information is checked automatically, and then it's dumped into the database and made more readily available to the executive committee of the ICTV that makes the decisions regarding what new taxa are going to be created. And Curtis Hendrickson in my group is working on the development of this taxonomy proposal management system. And then we're developing an application programming interface, an API, to make the information accessible in a variety of means, not just from the website, but people can do automated queries against the API to pull out that information. And then, of course, we're going to plan on having workshops, enhancing the documentation, having talks and in different forums in order to make it more available. So I'm going to stop here, and I'm happy to take any other questions that people may have.
Yeah, thanks for a great presentation. Yeah, and also about a very current topic such as monkeypox that you used as your example, and showing us some great resources. There were some questions actually that other people helped answer where to find things looking for Ebola virus. Jake had a few questions. Might be easier if you just read them, and they're kind of somewhat related. And we're just about out of time. So if anybody has to go to another meeting, we won't mind. But if you could stay for a couple questions, that'd be great.
All right. Let me see. Okay, Jake, how's the taxonomy of viruses different from the taxonomy work of other microbial organisms? Is it due to the large number of viruses and so on? I talked about that a bit when I compared it to the taxonomy of bacteria, in which you use a more simplistic method, that is that 97% cutoff to distinguish between different bacteria. But the advantage of that is you have one criteria that can be used across that whole kingdom of organisms. Whereas in viruses, we can't do that because the different viruses with different properties differ too much in, for example, their mutation rate. And so given the difference in mutation rate, the different features and characteristics that you use evolve at different rates. And so you have to define criteria differently on the basis of different families, different classes, and so on up the rank level. Can you give us an example of incorrect classification? Okay, what happens is as we get more and more viruses added to a species of, let's say, a particular family, and those viruses are then classified according to genera. One of the properties that's required for a complete evolutionary based classification scheme is that everything be monophyletic. So that is, if we have viruses classified into a single genera, that genera has to have a single lineage as part of the whole family. What happens, and this has happened a few times, is that when you add more viruses to that family and add more genera, what you see is the species within one single genus is split up. And one part of the tree has half the viruses or half species in that genus. The other part of the tree has the other half. Now it's polyphyletic. That tells you something's wrong. So now you have to go in and you have to look at the demarcation criteria. You have to look more closely at the properties of those viruses, find out the reasons why they're now polyphyletic, and fix it, and now make it monophyletic. That's happened. The bacterial viruses in particular have been problematic because of their high rates of recombination. And more recently, the ICTV has gone in and gone back to reclassify many of the bacterial viruses, and that's an ongoing process. So everything's not perfect. That's why the ICTV is always coming back and re-looking at the current classification. What's the most tricky taxonomic work you've done so far? I think in general, not just me, but the ICTV in general still has not had a good hold on how to classify viruses that are highly recombinant, like bacterial viruses. And that's difficult because you don't have, what you're looking for is one common conserved set of genes or set of sequences that's present in the original progenitor, the original virus that gave rise to everything that we now see. But with recombination, you may not see that. So that's a particularly difficult problem. Let's see, a question from Rita.
She was looking for info on Ebola virus, but then somebody else found it. So I actually had a follow-up to all these talks. I'll just talk about classification and taxonomy that, we know from evolution and mutation and divergence that it, like as Jim had mentioned, that it is a continuum, that it's a little bit arbitrary to say, oh, we have these distinct, kind of important named levels of classification. When you talk about like a clade, it's all like, continuous and almost infinite. So, and you mentioned that when you have to split something up, so, it just seems kind of messy, and how do you deal with all that? And related, I've been working, because I work on ontologies in this area, not just viruses, but also bacteria. that when you reclassify something, you have all this literature, everybody knew it by that name, and then the disease was caused by this thing. And now we know from genomic sequencing that they're not really the same, even though we thought they were the same based on phenotype. So, and now sometimes I see an organism have 10 or even dozens of names because maybe they look different under different conditions, but then after sequencing, you realize it's the same thing. So I'm just wondering how that, how people deal with that, how that everything being reclassified. Seems messy.
Let me answer the second question first, then I'll come back to the continuing question. So in that search box that I showed you when I searched for monkey, that has a check box, and you can check it and you can search across all taxonomy, all the whole complete taxonomic history. So for example, poliovirus, if you search for polio right now, you won't get a hit. But if you check that box and search across past releases, you will get a hit. And then every taxon that's mentioned in that listing has another icon you can check on for information, and you can get a complete history of that. So one way we deal with the changes is to provide the complete history of every species, every genus, every family, and so on. So you can investigate what's happened over the years and what's changed over the years of virus classification. So in terms of the continuum, so yes, it can be an involved process in order to determine what features are important to classify the viruses. But I would say that everything is not a continuum. There are actually discrete features that you can derive. But keep in mind that a discrete feature does not only have to be a discrete peak of nucleic acid sequence identity. A discrete feature can be made up of multiple criteria. So it might be nucleic acid identity plus common gene sets plus common order. Taken together, those may provide discrete demarcation criteria that then can be used to classify the different viruses. But in addition, if you begin to look at the sequence information, now this does change when you have more and more information, but you see, as I showed you, those discrete peaks in the pairwise sequence comparison graph that I showed you. And so in many cases, there do seem to be discrete peaks of identity. That is, if you think about it from an evolutionary standpoint, what you see are peaks of fitness and then valleys of unfit viruses. So it's like for SARS-CoV-2, we know that we have a peak of fitness in the ability to infect and cause disease from what we call the alpha lineage or beta lineage, delta lineage, and now the Omicron lineage. But there were lots of viruses that are intermediates there that did not cause significant disease. We never named them because of that, and they didn't evolve. They didn't continue to cause disease. So you have those peaks of fitness, and that's defined by discrete nucleic acid sequence similarity or identity in those different lineages of SARS-CoV-2. So evolution, whereas it might be considered a continuous process, in reality is not continuous, because not every sequence out there that's evolved during virus replication is as fit as every other sequence. And probably the vast majority of them die out because they aren't fit, because they can't replicate in the particular environment in which they happen to find themselves. So it's only those discrete peaks of fitness that are allowed to evolve. And that's what we try to find and define when making virus classification in the taxonomic sense.
Yeah, thanks for the clarification. And and we're never going to get all the information. It's never going to be perfect. We're not going to get every sequence every step of the way. So it's a model, so that we're trying to make it represent the real world in a way that's most useful. So there's always never going to be perfect. So it's biological systems. So yeah. So thanks for clarifying that. Any final thoughts or questions from anybody? I know people stick around till the bitter end, and even a few minutes past. Appreciate that. Well, if not, then you can always contact Dr. Lefkowitz by email. And thank you very much for the presentation. This presentation has been recorded, so we will be posting it, and it will be available after we finish processing it. Thank you, everybody, for attending. Thank you, Dr. Lefkowitz, and everybody have a wonderful weekend. Hope to see you at the next Power Talk.
Bye.
Okay, bye-bye.